the males on the top and the females going down the bottom. You can see the Y chromosome line going across the top. And we can identify the males in that line based on your Y chromosome because it will all be the same. Likewise, we can identify all of the females on the mitochondrial line that comes down across the bottom because they will all be the same. But at this level, that's 16 individuals. We've only identified 2 of the 16. And we've only analyzed the contribution of 1/8 of the ancestors of that individual by looking at the Y chromosome and the mitochondrial DNA. So you need to keep that in mind when you read and see a lot of the DNA studies that have been done out there on mitochondrial DNA or on Y chromosomal DNA, and understand that the picture that you are getting with that alone is a very small part of the picture. Okay? Because at the next generation, it's not just 1/8, it's 1/16. At the next generation, it's 1/32 of the information that you have. And so if you extrapolate that back 20 or 30 generations, there are some problems. Although it's not as bad as that sounds. There actually are some really valuable things that we can learn from both mitochondrial DNA and from Y chromosome DNA. However, this would be a hypothetical population structure if we look at the mitochondrial DNA. And you can see that there are a number of different types of mitochondrial DNA that we see in this total population. If you start up at the very top, you can see some individuals who have a red mitochondrial DNA, some that have a yellowish green up there at the top. And as we go down, you'll see that what's going to happen to the yellow mitochondrial DNA lineage? It became extinct. It was lost because the females carrying that mitochondrial DNA did not have female offspring. Okay? And what else happened up here? We had new mitochondrial DNAs enter into the picture with the purple as it comes down there, or the green that comes in over there. It can be a result of new populations moving in that carry alternate forms of mitochondrial DNA. It could be mutations within the red or yellow or green that could turn it into a different type of mitochondrial DNA. You can see that it's quite dynamic. And so as we go down and look at this bottom line, which represents the current population, we may if we removed all of the information in the previous generations, and let's just look at the last 2 generations, a generation and a parental generation, and looked at all of the mitochondrial DNAs that were present in that population, have we made an adequate survey of the ancient population from which these individuals derive? No, we haven't. Okay, we've been very limited in our observation of the population structure based on mitochondrial DNA, right? Because there have been a number that have gone extinct. Perhaps the sampling isn't large enough. It doesn't contain all of the DNA, mitochondrial DNA types in the population. So there's some real challenges in trying to reconstruct the past genetic patterns based on modern or present-day DNAs that are collected. There are some ways that we may be able to approach that. And this is one of the approaches that we've taken in our laboratory at BYU. I wish that I could say that this was the magic bullet and it was going to be able to answer all of our questions concerning ancient populations, but it turns out that the ability to recover ancient DNA and get useful information out of it is extremely difficult. It can be done, and we have been able to do that in a number of limited situations. But for the most part, it's going to be very difficult to go back in the past 1,000 years, 2,000, 4, 5, 10, 12,000 years and recover DNA from individuals and say something about their population structures. However, we have been able to do some of that, and it's reflected in this chart, in that we can go back and we identify mitochondrial DNAs in the past, that are in this blacked out section, individuals that may or may not have the same mitochondrial DNA sequences that are currently present in the population today. And this is particularly applicable to some of the Native American DNA studies. In just a minute, I'm going to talk to you about 4 or 5 major groups. We call them haplogroups of mitochondrial DNA, which involves A, B, C, D, and X. X was real troublesome because it was sort of an outlier for a long time. But it turned out that, and it was thought that it was, when X first showed up, it was thought that it was probably admixture from a European population. Something else coming in from somewhere else. It was not a Native American DNA. Until some of the ancient DNA studies came out, which identified the X group in a number of different ancient DNA samples that predated Columbus. In fact, predated Columbus by quite a ways, which said that there were more or different mitochondrial lineages present in the ancient populations than what may be reflected in the modern populations. Okay, and we'll come back to that. So there are a number of potential difficulties, although I'm a geneticist and I'm a molecular biologist, and like I said, I would really like for genetics to be the magic bullet, and I think in a lot of ways it really is, and it can be, and I'm a strong proponent of genetic studies and in helping us understand population structure and even ancient population structure, and that's what I spent the last 10 or 12 years actively pursuing. However, I think that it's very important that when we attempt to set up a situation such as has been set up out there, can we test the validity of the Book of Mormon by using genetic techniques? I think we have to be very careful about the hypotheses that we set up, that we actually set up a scientific experiment, a hypothesis that can in fact be answered, and that when we see the results, we can interpret those results in a proper manner. That, in fact, the hypothesis is set up as falsifiable, which means that if it's wrong, we can identify that it's wrong. Okay? Well, I'll come back to that. Second, it must make relevant comparisons. We need to be in the same frame of reference. And one of the most important frames that we're dealing about here is a time frame. The genetic information that we have has been on Native Americans today has been collected 99.9% of the genetic information in Native Americans has been collected from living populations today in the present configuration of Native Americans living on the American continent. Okay? You have to be very careful of making inferences about the past based on that, especially from what we know, what we think we know about past population patterns in the Americas, and I'll come back to that. I noticed in the conference last week that was referenced that there were some papers and some discussion on DNA. And I think there were some very good things that were said there. But I also have some concerns in that there was some mixing of the data across time scales that may not be appropriate. There's a lot of mitochondrial evidence that has to do with population structure very deep in the past, 10, 15, 20, 30, 100,000 years ago. And that the experiments that were set up to look at those were set up correctly to be able to answer those questions in those time frames. If we then extrapolate those to the very recent past, the last 2 or 3 or 4,000 years, I think we have to be very careful in extrapolating some of those results. As far as the populating of the Americas, well, okay, we'll come to that. We also be able to need to use the correct tools. Is it mitochondrial DNA? Is it Y chromosomal DNA? Is it autosomal DNA? Is it a mixture of all of those that we need in order to answer the question? I believe that it's a mixture of all of those that can be helpful in answering questions about population structure. What about the populations? Are we looking in the right places? Have we sampled all of the Americas? The answer is absolutely not. Do I expect the changes that I'll tell you about, or the